예, 이제 곧 시작하겠습니다. 밖에 계신 분들 들어와 주세요. Actually, we will begin in few minutes, so please come in and have a seat. 자리 세팅이 끝나는 대로 한 2, 3분 있다 시작하도록 하겠습니다. So we will begin the symposium in 2 or 3 minutes. So please have a seat. Okay. I think we are almost ready to go. And yeah, welcome everyone, and welcome to the first symposium on nanotechnology and biological systems. My name is Taehyun Yoon from Hanyang University of South Korea, and I will preside the sessions for this afternoon. And on behalf of the organizing committee and the sponsors of this symposium, I'd like to welcome all of you, and also thanks for your presence in this symposium. So to start this symposium, we have Dr. Ji-young Ryu from our national body, KACH, the Korean Agency for Technology and Standard. So please give a warm applause to Dr. Ryu.
Good afternoon, ladies and gentlemen. First of all, thank you for your attending to MBS 2015. I'm Ji-young Ryu, who is a researcher of Korean Agency for Technology and Standards, KACH, and in charge of ISO TC 229 nanotechnologies. I would like to ask your understanding on my opening speech instead of Director Jae-man Lee. He feels very sorry for his absence today. On behalf of the KACH, I would like to extend my warmest welcome to all of you visiting and participating here, Seoul, capital of South Korea. Let me say special thanks to Professor Taehyun Yoon from Hanyang University and Ms. Sun-Dok Kong from Korean Standards Association KSA, and Dr. Taegol Lee, who is a convener of study group on MBS in ISO TC 229 and Korean Committee of Nanovio and ISO TT 229. And I also thanks to all invited speakers today. I know you give us a wonderful talk today. And actually, I don't want to emphasize the importance of nanotechnology again today. Maybe you know already about that, right? And I hope you have a good time for sharing information, idea of nano and biotechnologies. And I also hope this is a good opportunity to contribute to the development of standard and technologies. And thank you again, and enjoy your time in our beautiful and dynamic Korea. Thank you.
Yeah, thanks again, Dr. Ryu, for opening remark. And as a keynote speaker of this symposium, I'm very delighted and very honored to introduce Dr. Simon Holland, who is the chairman of the TC 229 of ISO. Yeah, please come.
Annyeong, Seo. Good afternoon. I have to try, okay? Thank you. So each country I go to, I do try and use the local language. It was a bit of a challenge in Delhi last November, but I will try, so I'm sorry. That's the best I could do. I'd like to thank Professor Tae Hyun and his colleagues from the Department of Chemistry at Hanyang University for the invitation to give this keynote speech. I'd also like to thank the Korean Agency for Technology Standards for hosting this event, together with the Korea Research Institute of Standards and Science and the Korean Standards Association, and also Hanyang University for sponsoring the 3 days of meetings of the TC229 ISO working groups that are being held in this building. So, the title of my talk is Biotechnology and Nanotechnology Together. It's a nice broad title, sort of something you might get in a finals paper. Yeah. Biotechnology and nanotechnology discuss. What I intend to do is to talk about this in terms of standardization. I'm doing that because I think a lot of people in the audience are from the standardization field, and it would be a relatively easy reference. You could look at this at different levels, but what I'm going to do is actually talk about the benefits of standardization, applicable to both nanotechnology and biotechnology. I think I've stolen a slide from Steve Brown, one of the later presenters. So, Steve, I'm sorry about that. I then talk about how we deliver standards, or how we are delivering standards. And then, of course, the most important part of the discussion is to talk about why I believe biotechnology and nanotechnology are coming together. But first of all, I thought I'd tell you a little bit about myself, okay? I was interested in science at school, and when I was 16, the first exams I took were made rather easy in mathematics because we could use a calculator instead of logarithmic tables. So anyone who remembers log tables will probably know how old I am. So I studied chemistry at university, did a PhD as well, married to Julie, and last year I moved to the north of England after retiring from almost 30 years in the pharmaceutical industry. And I support Bolton Wanderers Football Club. You've probably never heard of this small team, but I'm a season ticket holder, and I take great pride in my team. So it doesn't look very good on me, but there's somebody you may know who's worn this. This was a marvellous midfielder. He stayed with us many years. His dribbling ability was absolutely astonishing. And then he left Bolton Wanderers for Crystal Palace. So, yes. And now I have more time to myself. I can see all the games, so there's no excuse anymore. But yes, I thought I'd just tell you a little bit about myself. So when I joined industry in 1986, I worked for the Beecham organization, and that was focused very much on formulation development of antibiotics. Interesting now, some almost 30 years on, there's an acute need for the development of new antibiotics. Resistance against antibiotics is becoming a real issue. And I must admit, there aren't many pharmaceutical companies that are looking at this. So I used to work with antibiotics, so this was sort of traditional medicine. I managed to launch a product in 1994, one of the very few I've done. And then the Beecham organization became SmithKline Beecham, and I moved on from penicillins onto neuroscience drugs. And this is where I was introduced into the field of submicron or nanotechnology. So what we actually did is we made material that was just a couple of 100 nanometers in diameter of active drug, embedded that into a tablet or a capsule, and I actually set up manufacturing sites in Scotland and in Ireland. We actually got the very first license from the Irish Medicines Board, which is the regulatory authority in the Republic of Ireland, similar to the FDA, to make a drug product intermediate of nanoparticulate material. I was really proud of this. I took 100 molecules through development. None of them worked because they failed in the clinic, but that's what happens with pharmaceuticals. And then later in my career, when I went to GlaxoSmithKline, I developed respiratory products. So there's 2 products on the market now, Relvar Brio and Anoro. I was also involved in change control for 6,000 staff. I really wouldn't recommend that to anybody because it can be a full-time job in itself, trying to control change. So really that's just the background to my introduction into nanotechnology. It was from pharmaceuticals and exploiting the unique properties of drugs when they get to the nanosize scale. And I had a marvelous time developing those products. The big challenge for us was actually characterization. So if you make a nanoparticulate drug particle, you then put it into a formulation, how can you show that you can consistently get the same particle size distribution of that drug into the body, in the stomach, if it's given orally, throughout the shelf life of that product? We had to do an awful lot of development work with laser diffraction and other techniques. It was quite a challenge, I must say. And in the end, there was no guidance. We had to speak with the regulators, say, this is what we've got, what do you think? So it was a challenging time. And here in 2015, I don't think it's changed. If you've got a new application with healthcare, you should really speak to the agencies because they're just as interested in what you're doing as the rest of industry is, and you've got confidentiality. And the more you share with them, the more likely you are to get a positive response. If you go in rather defensively, it won't work at all, particularly with the FDA. So, this is the slide I've stolen from Steve. And really, I think I'm preaching to the converted, okay? This is really important. I think it's self-evident, all these benefits from standardization. You share what you need to do to establish best practice. And with a lot of the standardization work that takes part across different platforms, not just ISO, it's a multinational approach. It also helps to encourage development of new technologies because there are some external references to help you. And I think if you've got stakeholders to convince that you're actually doing a good job, if you can actually refer to an international published document contributed by many, it actually helps communication with stakeholders. Rather than just saying, I've got this wonderful product, this is what I'm doing, you can actually say, actually, there's something external here that's been peer assessed. In theory, you can develop products that meet recognized standards, should make trade easier. Now, that might not always be the case, but it should lower barriers. And of course, a big aspect I'll come on to in TC229 is that by having standards, you could actually ensure that we help protect public health and also the environment by controlling what we do. And of course, if standards are very good, you can actually help then to shape what the regulators might want to do in your own country and across countries because they're recognized as being a very high standard. They do take a long time to produce. As many people in this room will know, it can be several years to achieve a standard because of all the rigorous interrogation we have. As we've noticed yesterday, today, and also we will see tomorrow in the working groups that we're taking part in here, thanks to your generous sponsorship. So these are just self-evident benefits. I don't think anything I've said here is new to anybody. How we deliver them. This is just a summary of where we are with the ISO TC229 Nanotechnologies Committee. When I took this role on as chair, I didn't realize how big the group really was. There's 33 participating countries and 15 observing countries. At a full technical committee meeting, we can have anything up to 160 delegates. So it's quite a big group. The Secretariat happens to be the United Kingdom at the moment. And what we have are 4 main working groups. So obviously we have Joint Working Group 1, which focuses on terminology and nomenclature. So it's great if you have the terms and the definitions. Then you need to find ways of measuring things, Joint Working Group 2. Health, Safety and Environment, Working Group 3. And then a new group, a relatively new group, that I'm participating in discussions here, is looking at material specifications, Working Group 4. On top of that, there are two very important additional groups. We have task groups on nanotechnology and sustainability, and also consumer and societal dimensions. That's quite an important part. If you're taking a new technology forward, you need to think about the impact on people and actually address what we're doing on nanotechnology in terms that the general public can actually understand, rather than dress it up in a language that isn't particularly easy to understand. And then at the top, we have the most recent group, and the group that's actually the reason why we're here today is we have a task group on nanotechnology and biological standards led by Tay here. And that is an emerging part of the work of TC229. And I'll try and explain later why I believe this is really important as I try and talk about our relationship from nanotechnology to biotechnology. So we've published 42 standards. There are 25 in development, which is quite a reasonably productive ISO group. I know you've all got this printed already, so you can probably see it. I'm not going to read all this out, but it's just saying what we do. So the reason I'm showing a picture of all the beautiful people, this one's from a meeting a while ago in Mexico, is that the decisions of the ISO standards group are reached by consensus, not by a vote, but by consensus. Now, in doing that, it requires a very rigorous discussion on what the subject matter is amongst the experts, but it also takes a lot of discussion, and I've often seen what I call violent agreement in a room where the dividing factor is the common language of English, because people have expressed exactly the same points, but not quite realized that actually everybody's agreeing. And eventually the penny will drop and people say, yes, I've got it. But I think by the fact that all these documents, the standards that are published, are reached by consensus, means that they've really had a good run around. They've not been forced through a committee, they've not been voted through, they've been a jolly good discussion that leads to a peer-reviewed outcome. And people that all smile all the time, you should come along to some of the working groups I've been in with sometimes scratchings of head. Yes, this is one of the bigger groups, and I'm really pleased that we're able to hold these interim meetings here. We always rely on hosts to help us with the meetings, and it's good that we can have interim meetings because of the sponsorship here of all the different groups. So, just to pick up on a couple of areas of current focus, you've probably all heard of the wonderful material called graphene. This is a typically British material, discovered in Manchester, patented elsewhere around the world. But what's important is that we work with not just within TC229, but there's the corresponding electrical group, IEC 113, and we're actually developing technical specifications across the board. I'm not saying it's easy, because each group, so the electrical group, the IEC group, have their own ways of doing things, and there have been disagreements, but graphene's a very important material for the future, and so it's important that we start with the absolute basics to get it correct, things like blank detail specifications. So that's one area of focus that you may have heard about, but more importantly is the more recent one, which is the nanotechnology and biological standards. So at short notice, we asked Tay to start off study group, and we had quite a fun time in Delhi, where I can't remember how many people fitted into that small room, but it was the hottest ticket in town, Tay, wasn't it? And we had to decide what we wanted to do. Now, prior to this, and it relates to my experience in industry, we did try and come up with a few nanomedicines definitions. because you've got to be careful what you actually describe. So there is a small technical report that we've issued. But we had to then think, if you're going to set up such a study group, how could we integrate this Duke group with a TC229? So I'll come on to the what we're actually doing with the NBS group later. But I thought I'd give you a bit of personal insight into why I believe we deliver standards, because I worked in product development, delivering new products for a long time. And I think standards underpin product development. Some people may say they hindered it, but I disagree. I think they actually underpin. So they support, they help form the basis of justification. So I tried to produce this structure from the bottom. So if you have a common foundation adopting standards, you've got a good way of comparing and controlling new products. And if you do that and you publish the standards, you actually start to build up confidence in emerging technologies such as nanotechnology. And also people start to ask how you manufacture the products, and I believe you get better interrogation of process robustness, how you actually make things. And certainly in the field of nanomedicine and medicines in general, how you actually control the manufacturing of these materials is becoming really key. So the fancy term at the moment is control strategy, but quite frankly all that means is how do I control the manufacture of materials? And if the regulators can see that you're following standards, it doesn't have to be the ISO platform, it can be others, it actually helps build confidence in what you're doing. And it's a bold step, but I actually think that because of the transparency of what we do in standardization, we can build up a bit of trust with all the people involved. So it's not just the regulators. If you've got a new technology, you've got to sell this to everybody. And it's from the general public all the way through different levels of government, of course, all the way to the regulators. So I think adopting standards actually helps underpin what we do in industry. Now that's a personal view, it's not an ISO view, but just looking at it from a distance of what I used to do all those years ago, I think it really helps what we do. Now, what you're really interested in, why do I believe nanotechnology and biotechnology are effectively becoming closer to each other? Well, in simple terms, if you look at scale, and what you have across here, if you can't see very easily, are powers of 10 of nanometers. The grey part is, sorry, I beg your pardon, if you go from 1 to 100 nanometers, that is the approximate dimensions of nanotechnologies. It's not limited to exactly 100. Nothing magic happens at 101 nanometers, but approximately 1 to 100 nanometers is the area in which the nanotechnologies arena is set. And of course, if you look at sizes are adjacent to that, then you have actual biological entities. You even have biological entities within that scale, such as antibodies. But bacteria, 1,000 nanometers, and viruses, a couple of hundred nanometers. So on this little edge of nanotechnologies, you've actually got real biological systems that are working a little bit higher dimension-wise. So I actually honestly believe that we're seeing overlap between what's traditional nanotechnology, manufactured materials, and what's happening in real biological systems. So it seemed to be obvious to me that it's inevitable that the two technologies would actually come together. Because people are looking at real applications, what's actually happening in cells. People have targeted drugs to cells for many years. If you target nano-sized material into cells, well, I think you're in the field of biotechnology and nanotechnology. Now, if this is the case, of course, where does the research sit? Is it actually fitting into the healthcare applications? If so, which standards then do you have to follow? And more importantly, which regulations do apply? So these were some general areas that I thought about. But what I really wanted to do above all is to stop a reinvention. So we're in our little field of nanotechnologies. There's a biotechnology arena. And what I wanted to do was to take common terms from each of the areas. What I don't want is to invent new terms to describe biological activity at, say, 200 nanometers. If there are things already out there, because I know how rigorous it is to actually get discussions on terminology, I believe if moving forward with Tay's group, we should see what we have between from already existing in the biotechnology field. I don't like reinvention, and I think it's important that we should actually try and adopt what they have. Now, to do that, we actually need to initiate a discussion between the two areas. And this is just taking the definition a little further. This is a definition of nanomedicine from the Food and Drug Administration. So it talks about nanotechnology, again talking about being approximately 1 to 100 nanometers. And then nanomedicine, the use of nanoscale materials for medical applications. Now, to me, the scope is really broad there. You're outside of 100 nanometers. All sorts of new areas are covered by this. So nanomedicine is one example for me of where biotechnology and nanotechnology are already working together. Now, as we develop nanotechnology and biotechnology, there are specific challenges. I think I'm preaching to the converted, saying that you needed to establish validated measurement and characterization, not just the equipment, but how you do this. I remember in industry, it was quite hard to get an analytical method to work in the United Kingdom and also in other countries. Exactly the same method, but trying to get it to work the same across countries can be difficult. And when you have specialized technologies to characterize nanomaterials, that makes it even more difficult. Of course, a really important part is to improve our knowledge about the impact, not on just health of people, but also the environment when they're exposed to nanomaterials. And of course, that's being addressed through Working Group 3 in TC229. And a lot of what's being carried out in nanotechnology is still pre-commercial. There are nanomaterials in commercial products, but it's still evolving. And of course, if we want to make nanotechnology standards work, we need to have active participation from the companies that are developing these products. So not just from nanotechnology, but from biotechnology groups as well. And I do encourage people from industry to try and join standards groups like ISO TC229. It's a big commitment on their behalf because they've got their day job. They've also got to try and help out with the standards meetings as well. Now, that was nanotechnology challenges. What about nano and biotechnology challenges? Well, the scale, of course, is broader. It's not just 1 to approximately 100 nanometers. Then I've already mentioned I want to accommodate the existing biotechnology standards into nanotechnology standards, and I want to avoid reinvention. And I also would like to think that we could develop reliable characterization methodologies at this intermediate stage between nanotechnology and biotechnology. Now, of course, you mentioned nanotechnology or biotechnology, and there are all sorts of concerns in the general public. The picture is of a young Johnny Depp in the film Transcendence, where, if I'm correct, they used nanotechnology to rebuild an individual from a computer program. And those sorts of images can cause concerns in the public. Who's controlling this nanotechnology? Now, this is science fiction, but people think, ooh, this is going to be quite scary. You had all the nonsense about nanotechnology leading to grey goo across the world as nanobots destroyed everything. The thing is, this sort of thing persists, and we've really got to overcome these concerns. There's actually a company in the States called Nanobaio. Of course, there are many nano products out there, like nanostat emulsions. So when science fiction like this comes up, there are often a lot of unanswered questions. But I'm saying that by application of standards, we can actually address those concerns. Particularly with the enhanced graphics you now get in these sorts of films, it all looks as though it's done like that. And people expect that's what people will do in the future. But I put unfounded because I believe there are unfounded concerns, but they will come up. And that's why we have to have vocabulary that's robust enough to deal with general queries from the public, not just from the scientific bodies and the regulatory bodies. We need to to work together to make sure that when discussions come up about the nano-bio interface, we're actually coherent enough to actually talk about this in a sensible way to the general public as well. So I've talked about TC229, which is the Nanotechnologies Committee. We have a sister committee, ISO TC276, and I think the best way forward, and we'll talk about this I'm sure a lot later with Tay, is that we need to reach out to see what existing resources we already have. So TC229 is nanotechnology, TC276 is biotechnology, and they're the chair and secretaries and the ISO Secretariat contact names. We are fortunate in that Laura actually looks after 229 as well as 276. Now 276 is quite a modern group. There are quite a lot of participating countries and observing countries, but they've not published a standard yet. They've not published a standard. Unlike 229, where there are many standards published, they're still working on these. So looking a bit more at 276, a group we can work with, they have similar working groups, not identical of course. So working group 1 talks about terms and definitions, 2 biobanks and bioresources, 3 analytical methods, 4 bioprocessing, and 5, which is very new indeed, talks about data processing and integration. There are also other groups mentioned at the bottom of this slide. So what I've been thinking about is how can 229 and 276 work together. Now if you look in the business plan, I don't expect you to look at this, it's very busy indeed. But this is the workflow within 276, and gosh, that looks very busy. Their business plan is quite heavy as well. So what I tried to do is to distill out of that what we could do. So in black, you've got the biotechnology group, the individual working groups, and in green, if it shows up on the screen, okay, where I believe we can actually link across TC229 from nanotechnologies across to biotechnology. So terms and definitions, gosh, that's quite an easy one. It's joint working group 1 from TC229. Biobanks and bioresources, well, Tay, I think this could be an area for the NBS group. Analytical methods, again, this is metrology, so to me, correct me if I'm wrong, TC229 people, I think it's a nice fit with joint working group 2. Bioprocessing, again, a question mark. This might be another area, Tay, for the NBS group to look at, as with working group 5. And a lot of what's in the other parts, because they use the term metrology, I think would be a good fit with joint working group 2. So what I did is I've mapped how the committee I work on could work with TC276. Well, that's all nice and academic, but how are we actually going to do it? Well, before this meeting, thankfully, I actually spoke to these guys. So on the 11th of May, I said, perhaps I want to, it'd be helpful if I actually explained what we're doing in TC229 to Dr. Gent. And I'm pleased to say we had a very productive meeting. It would have been awful if I'd come here and said, oh no, we disagreed on everything. But yes, so 2 weeks ago we had a good discussion. They've actually got a terminology compendium, which I've not received yet. So it's got all the definitions they're working on from biotechnology. So as I said, we'll take that and we'll look after those and see how we can use them. 276 weren't aware that we already had definitions bio-nano and nano-bio. Didn't know. It's amazing what you don't realize what you have. So I've shared that. Now they were invited to today's meeting, but it was short notice. But they are going to send a delegate to our next 229 meeting in Canada in late September. And of course I then got the corresponding invite. Oh well, perhaps you can come to Tokyo in October. Well, maybe. What are you doing, Tay? Yes, so really what I'm trying to put across is that I think a way forward, if we're going to bring biotechnology and nanotechnology together, certainly in the field of standardization, is we should work with the partner group, TC276. They don't have equivalent group to environmental health and safety because a lot of what they do in biotechnology is actually through established regulation, so they don't need to go there. However, when I actually talked about consumer and societal dimensions, they were really interested in that. Apparently they've got an ethical box that they have to tick on everything they do. And they were interested to see that we have very skillful lawyers who are part of our group who may be able to assist them. So again, we weren't aware of what each other had until we actually shared them. The only thing they told me, and I think there may be people in the room smarter than me, is that please avoid the term synthetic biology because it doesn't actually exist. Okay, I said, fine, okay. And we also will have to be careful with the term nanobiotechnology because it actually has specific meanings from a regulatory and legal perspective. Now I want to develop that further and see what they mean by that. So I think we're helping to set the scene of how we could take the nanotechnology and biological systems task group further and make it a much better and bigger group. So I was asked to present on biotechnology and nanotechnology together, question mark. Well, quite frankly, I think they are a good fit. And my message to you today is that I believe there's a clear relationship across the groups, and I can see a way forward by developing the TC229 MBS group and learning from other groups such as TC276 and other standards groups. It doesn't just have to be ISO. And then trying to understand the language from the biotech side, and they'll have to understand what we do. So I honestly believe, ladies and gentlemen, that there is a clear relationship between them, and this is an emerging field that I think we need to address, and I'm glad we've made starts to this already. So thank you very much. Any questions?
Liaison with a number of other TCs in the medical area and healthcare related areas.
Yes, Jeremy, you're right. I've picked on one specific one, TC276. There are other areas that we need to talk to. The 229 organisation has an enormous amount of liaisons already, but I'm hoping to speak with Dr. Gent to actually establish what the best way forward would be there. What I want to avoid is to reach out to, say, 10 or 12 different groups and end up with a very unclear outcome. But as soon as I use the word medical, then that has all sorts of implications. So I think I'm going to suggest that TC229 starts with biotechnology. I'd also suggest that we actually look at the non-ISO groups as well. Now that's a bit of heresy with my ISO hat on, but no, I think we should just talk to all the other groups. It doesn't have to be exclusively one platform of standards. And Jeremy, if you've got some specifics that you think I should be prompted with, then you can tell me a bit later. Good question. Come on, don't be shy. Ask me anything. Oh, come on, Ali.
To 100 nanometer due to the toxicity issues. Sorry. Do you think this could be possibly in some way modified, the specific size assigning for nanobio area?
Yeah. Well, the strict definition, Ali, of course, in the nanotechnology side uses that amazing word approximately. From 1 nanometer to approximately 100 nanometers. And what I put up earlier in the talk is the FDA version of nanomedicine. What I did when I worked in industry is I titrated biological effect against particle size. Okay, so if you don't need to go to 50 nanometers, you wouldn't. And actually that became really important with what we were doing with the medicines, is that we had to show in a clinical environment what the effect of particle size was on the clinical effect. And trying to make material of different particle sizes that all looks the same and control it in a clinical environment was an absolute nightmare. We had to use different colored capsules because it all looked the same. So I think what we do is the term in the nanobio area doesn't have to be 1 to approximately 100. I think if we have some specific examples, Ali, we should break this upper barrier, because it's not just nano, it then becomes, well, in current terms today, NBS. So we should not be so rigid, because we're actually acknowledging that it's outside of nanotechnology, and it's within biotechnology. So I'm quite happy to break this rule, although, as I remind everybody, that lovely word approximately has always been in the definition. Thank you for your question. Of course you can.
Nanovio, like in terms of TC229, I think you have some slide.
Yeah, I knew this question might come up. So what I've pulled up is, now this was a couple of years ago. This wasn't, it's not absolutely up to date. This was some thinking about 18 months ago, if I'm correct, Steve and Ali. And we had a go. So nanobiotechnology is the application of nanoscience or nanotechnology to biotechnology or biology. And the reverse, bio-nanotechnology, is the application of biology to nanotechnology. In other words, the use of biological molecules in nanomaterials, nanoscale devices, or nanoscale systems. And I was really pleased that we wrote that down, because when I spoke to the biotechnology folks, that was one of the first things they asked me. And then I shared this, and it was like, oh, right, oh. And then they threw back, well, yeah, but watch out with synthetic biology. Don't talk about that, it doesn't exist. So I'm really pleased that we've got something there under the technical specification 80,004/5. So we've got a start there. But what might come out during this afternoon is more discussions about what nanobio and bio-nano is.
Yeah, actually I was wondering like how the TC276 people think about like these terms, and if they agree with us or.
Okay. Well, where we are is I've given them this, and they're meeting on something like the 8th of June, and they'll come back to me.
Okay.
And I'd be surprised if they either fully agree or fully disagree. I'm sure there'll be somewhere in between. But do you know, Professor Yoon, this actually helps start the debate. I think it really helps with when I'm approaching someone else. I said, this is what we got, but what do you think of it? Rather than me imposing this on them and then saying, oh, no, it's not acceptable. So I think it's a useful way into the starting point between
nanotechnology and
biotechnology. Yes. Yeah. Now I'm sure we're going to hear other talks this afternoon, more details about what we have, and I know Steve's got some excellent suggestions of how we can take MBS forward within TC229. So I'm not going to steal his thunder in that. But yes, this is an interesting initial definition.
Yeah, I think we can also discuss it during the panel discussion time.
Yeah.
Okay. And is there any more questions? If there is no, then let's thanks to Dr. Simon. Thanks. Okay, then let me move to the next speaker. Okay. While changing the presentation materials, I would like to introduce the next speaker, Professor Taek-Hwan Hyun. He is a very distinguished scholar in South Korea as well as in the academia of the world. And he's a director of the Center for Nanoparticle Research, which is one of the Institute for Basic Science. And he's a professor at Seoul National University and associate editor of the Journal of American Chemical Society. And he will present about the designed chemical synthesis and assembly of uniform-sized nanoparticles for medical and energy applications. Yeah.
Thank you very much. Yeah, good afternoon. First of all, I have to thank Professor Yoon and Dr. Lee for the kind invitation. Actually, I'm not expert in the standardization, but rather I'm actually using the nanomaterials, synthesis and application of nanomaterials, in particular uniform size nanocrystals. That's what I'm going to talk about this afternoon. In particular, for the recent, medical application we have tried in our laboratory. Okay, so for the last 17 years in Seoul National University, I've been working on the synthesis of different kind of nanomaterials, in particular uniform size nanocrystals. And then more recently, I'm interested in the applying proceed.
Okay, we have two more speakers before the coffee break, and our speaker for this time is Dr. Steve Brown from USA. He is a co-member of the ISO TC229, especially the Working Group 3, and he will present about the development of bio nanotechnology standard in ISO TC229 nanotechnologies. Pause forward. Yeah.
Great, thank you. Well, welcome afternoon. I'm glad to see you're all still here. For those of us with the jet lag, it's actually a real challenge in the afternoon. So, and also I'm going to admit right now, my paper will not be nearly as exciting as that previous one. That was just incredible work. As we become older, we start knowing people with cancer, and to think that we have such intelligent scientists working on these solutions, it's really, it gives me great hope for the future. So I applaud their work, and your else's. So the title of my talk is really how do we go forward, and somewhat building upon Simon's comments of what's our next steps. So you've just heard a nice talk of scientists that are working in this field. We want to do everything we can to help support those type of activities because they're so important. My presentation will somewhat be in 3 sections. One, just an overview of how we develop standards, and this will be very quick. The second one is kind of our scope of what TC229 is. But more importantly, what I want to focus on is what is our proposed path forward. We have a meeting coming up later in the year in Edmonton, California. Edmonton, Canada. Good thing my Canadian friends aren't here. They would be booed out of the room. Right. Yeah, there again, apologize for the jet lag. But really, we as an organization, what do we have to do to move forward so we can help in the standardization of this important field? So similar to Simon, we talk about why these standards are important. Communication among stakeholders. We heard the example of the simplest definitions of bio, nano, bio. What do they mean? And if we're going to write documents and material specifications, we have to be able to communicate the same language. And particularly, we have a unique role that we have a really excellent group and staff that actually can develop the nomenclature. We have a large amount of input from lawyers, and they are very specific about writing down exactly what the definitions mean. It can be very tedious listening to them talk because they will argue actually for hours over the meaning of approximate, right, and include those. I look at this once again as lower barriers to market entry. You know, if we can all understand what we're doing, it reduces our cost to all the organizations. So one of the things, and I think this is real important, you know, what are the factors that drive standards development? First and foremost, you have to have someone propose, here is an idea. Here's an area that's open for standardization. And that comes from our national body members, and they are volunteers. There is certainly biases, there's certainly desires. So if one national body, if they are interested in a particular industry, they are more inclined to be active members and start putting forward new work items. But ultimately, we need individuals that have an interest that are going to put forward something, and whether it's an economic interest or you know, unique to their area, we personally don't care as long as it benefits all of us. One of the things we try not to do is to be so esoteric that it only benefits one of our members. So that's why we have a balloting process to try and even that out. Another thing, and you'll hear about us talk about, we do surveys to try and find out what industry needs. You know, the iron that we just talked about, you know, is there analytical techniques that industry needs us to help develop? Specific requests a lot of times are liaison members. You heard Simon talk about reaching out to 276 and what do they need, and we may have the expertise, so if they can let us know how we can help further their activities. One of the things I really point out, I think it's very important, this last bullet, it's really driven by stakeholder input. And if we don't have the right people within our organization, we could be developing standards that aren't necessarily valuable. Simon gave a great insight on his background in the UK and his love affair of soccer. One of the things I look at what I can bring to this conversation is I've actually been at every meeting of ISO 229 except for the very first one. So somewhat from a historical perspective, one of the things that we somewhat did wrong in the beginning is we had lots of people that had very special interest, and we developed some standards that you look at them, they were not as relevant, and we could have spent our time better developing standards that everyone benefits. So it's very critical that we find out the stakeholder needs, and I view stakeholders as industry, academia, other scientists that we're hearing presenting today. If someone came up and said, I need an imaging contrast agent technique, that helps us really focus on what we need to do. So initiation, there's different types. We call them new work item in process, NWIP. They can come from a creation of new standards, revisions of existing, modification of existing programs out there. They come from, as I said, many sources, internal, external agencies, whether it's regulatory, whether it's academia. We're essentially open to anything that a national body wants to propose. So I think now I want to really, when I introduced about saying I've been here since the beginning, this is our current scope. And it really, it's easily 10 years old. And if you think back 10 years ago when we thought of nanotechnology, our primary focus was magically 1 to 100. Well, we now know there's nothing magical about 1 to 100. It's a nice set of numbers. We could have said 1 to 10, but at that time, we somewhat knew that the quantum effects were somewhat related around maybe 40 to 50. We now know there's all these special effects. We've heard of people talking today, particles down in the 2 nanometer range. So this number of 1 to 100 is arbitrary, but there again, this is our definition of our scope. Two primary interests. Number one was 1 to 100. The second one was unique properties. Right? Who can define unique? And what is unique to me, if I'm interested in cadmium and its fluorescence, someone else may have no interest, and that's not a unique property to them. So you can somewhat see we did a very good job 10 years ago, but it's naive when we try and say that this scope is still applicable today. And I think Simon mentioned this, within this scope, we have these the 4 groups with terminology, instrumentation, EHS, and product specifications. But as we go through this talk, I think that's one of the things that you're going to hear me talk about. It may be time that we actually revise what's in this scope document. In case anyone was noticing, nowhere in the scope does it say even the word biology, much less bio-nanotechnology or nanobiotechnology. And in fact, 10 years ago, that concept didn't even exist, right? I mean, just that frontier, we've learned so much in the last 10 years, and the industry and our research has progressed so much faster. I won't spend a lot of time on this, as Simon's already displayed. This is kind of our organizational structure. And the challenges. And these are very real challenges, and we need to really comprehend these because they determine if we, in fact, can bring something in for standardization. The early stage of development. the times in the past where we have tried to develop a standard when it's somebody's research project, what happens is it takes us 4 years to develop that standard, right? I work for Intel. Our market time is in 6 months. 4 years is incredible. For me to go back to my management and say it took us 4 years to develop a document, they're like, why are you participating, right? So it's very, very important that when we have new items in this early stage of development, we're looking for, it's already been tested in the laboratory, it's already been vetted, we've had round robin, we have quality assurance, and now we're ready to standardize it. So there again, we have to be careful, and this is a hindrance. And in kind of the bio-nanotechnology, we have to be very cognizant of what we want to pull in. Lack of active participation by bio-nanotech manufacturing organizations. And I applaud Ty for this symposium, because this is part of our outreach. And the more that we can talk to these other outside and saying, this is capabilities, this is what we can do, and we're here to help serve and help your development of these great technologies. The last one, it's another challenge for us, but as part of ISO, we are responsible for interacting with these other technical groups. And once again, Simon is showing great leadership by going out and saying, I'm going to have an outreach with another group within ISO that actually is very closely aligned. If we don't do that outreach, they will go down their path, we'll go down our path, and we have duplication, and we're not as fast as we should. Thank you. So really, this is somewhat of a proposal. There's 10 steps, and I'm hopefully, even though I did drink a lot of soju, I'm not an alcoholic, so this is a 12-step process, but it's only 10, and it's really more related to just the standards development. And just a warning for you, those of you new to create, that soju, if it's very cold, it tastes like water, but it is not water. So just that's what I learned from last night. So the steps, the first one is we need to look, and once again, do we need to modify our scope? And I'm going to go in these details just as a quick overview, but really determine what are the standardization needs. And if someone were to come back and saying, we can cure liver cancer, if you guys can develop these standards, we will, trust me, we will change all of our focus. So we just need to know and have input and make sure that we're working on the right items. Evaluate the state of knowledge. Identify, the rest of these are kind of our standard process, but how do we get it into our roadmaps so we can advertise, recruit, active recruitment. I know from within the U.S., within our technical advisory group, organizations, they're busy. People are busy. These are volunteers. You have to go out and actually find these individuals and convince them of the benefits of participation. They will not just pick up the phone and call you. In fact, most of the time they will hang up the phone when you call them. So you have to have a very compelling reason of why and what are the benefits for them. And then we have a standard process by which we develop the standards. So let's talk about the scope. I had this slide up before. And the basic question, and we've been grappling with this for now 6 months, and I think it's time that we actually make some concrete steps. But, there's an argument why we should modify, because it helps within the ISO world of everyone knowing that, hey, our TC229 and nanotechnology, it includes bio applications. It actually aids when we're out advertising of saying, hey, here is an active committee that was working in fields that you may be interested in. The counter argument, and I'm not trying to weigh the pros and cons, but we've had a counter argument that we don't have graphene in here, right? And the graphene specialist may look and saying, I think graphene's the most important, we should include that. We don't have carbon nanotubes or multi-walled carbon nanotubes. So that is somewhat of a different that we'd actually be calling out specifically. I don't personally, I don't think that's a reason why we wouldn't want to change it, but you just open up that scope. And purposely, I think that one of our things that we did right 10 years ago, we did leave it broad. So we've had all these different types of standards. But so we may be at the point of, if we can narrow the focus, if we can start putting in these additional terms, it will help us recruit new members. The similar slide that you saw from Simon, where it has the actual definitions of what these work groups. So not only do we have to modify our larger scope, but once again, you don't see any mention of bio-nano in any of our current work groups. And this is very important because as new standards come in, we need to evaluate, is this within the scope of the metrology? Our next one, in the step 3B, we talked about a survey. So we have conducted surveys in the past. I know a number of us in this room, we actively participated. But I'm almost going to say it was a survey done by volunteers. And I think we're almost at the stage of, if we do this right, we have to have, not to say that volunteers do really good work sometimes, but we almost have to have a very professional, very focused of saying, of a survey, of going to like an organization like the FDA and saying, is there work in this field that we can support you? And I have no idea, but if something like the FDA or another country's FDA comes back and says, we do not want any ISO involvement, we should learn from that. But it's only by having a very focused survey to actually go off and kind of answer those questions. Our previous surveys, I know within the US, we send out the survey, maybe you get a 20% response rate. That is not going to be good enough. We're going to have to be very active so we can have a really good focus on what our future for the next 5 years should be. I have, and I've mentioned it before, kind of the outreach. It has to be very, very active. And to me, we have to have people assigned of saying, yes, you're going to talk to the medical device companies. You're going to talk to the imaging. There's a lot of instrumentation. We just heard of MRI. Is there an MRI company that we need to be talking to and saying, how can we support this biological applications for MRIs? Evaluating the state of the knowledge. Once again, it goes back, are we actually ready? Now, sometimes we're not ready, we don't have a standard that's already been developed, already in a laboratory, we can have other ones of technical reports or kind of the state of the art, and we can somewhat make recommendations of saying, this is the areas, this is what we currently know, and these are what our gaps are. So even if we're not at the stage of bringing a document in for development, we can still aid in that process by what we publish and what we put out in literature. And there again, kind of for a true new work item proposal, it has to be based on something that's existing in the literature. Step 3D, based on the survey results, a lot of this now is somewhat perfunctory. We start modifying our scope. We start, trying to focus us. Really, kind of, this is what the survey says we need. These are the industries that we're targeting. And we're not trying to solve world hunger. We're trying to find a particular group of standards that we have the expertise that we can go work on. And I've been told more than once in my career is like, Steve, don't try and boil the ocean. Identify what you want to accomplish, and let's focus on that. This is kind of the complex world and that we live in ISO. All of those little circles either represent ISO technical committees. I've also put up other organizations like OECD, outside organizations. And really, what we need to do is, after we modify our scope, we need to pick very targeted and saying, these are the ones that we want to be active in. And once again, the outreach to 227 is a primary example. Medical devices, they have lots of applications in this field, but they may or may not need support from us. And so to me, it's better to find out that early, so we don't have duplication, or in fact, I look at it worse than duplication. Here's a need that's going unfulfilled. Going back to our roadmaps, our roadmaps are really try and say, these are the areas that we want to have standardization. We have turnover, new people coming into 229 over the last 10 years, probably at least 50%. If we have good roadmaps, new members that come in, we can put up the roadmap and saying, can you go back to your country because this is the area we're interested in? Do you know of anybody that has expertise in imaging? Do you have any equipment manufacturers that are looking at that cell wall interface? So it's a very targeted on our roadmap, and it keeps the extraneous work items. When they pop up, we say it's not on our roadmap. We're not going to focus on that. Kind of the promotion, the very active promotion, we cannot sit in a vacuum. If we don't have a targeted outreach, we're never going to bring those experts into this group. It has to be very focused, and we have to have a compelling business reason why we want to bring them in. So you can start telling, I've got this Intel speak in there. It's like, what is your compelling business reason? What's the ROI? And how do we get these, how do we make progress here? And I apologize for that. But I do think we need to take somewhat more of a business approach in the standards, because it's a very time sensitive. And once again, I'm just blown away by our previous talk about here's potential cancer agents, fighting agents. What can we do to support that type of activity? One of the things that we have done within the U.S. is to develop a communication package that any one of our members in our national bodies, we can go out and we call it the elevator speech within just 5 minutes. These are the reasons why you should participate in standards development. And to me, that's the only way. We've got great participation by standards development bodies, but they're primarily government organizations, and that's kind of their mandate to participate. Right? This is more looking on those private industries or the academia that are involved in this work. Once again, kind of the outreach, a very targeted outreach. And Ty, as we talk about our study group, to me, once again, it's got to be very focused of saying, here are the individuals, these are the industries, these are the people that we need to target and go after. There again, we can't have everybody just put out a flyer that no one reads. It's got to be a very targeted kind of a sales force. I'm going to make cold calls and get people to sign up. Once again, this is somewhat perfunctory how we do business within 229, but we do have a checklist. And if your new work item proposal doesn't meet that checklist, we're going to dissuade you from submitting it. I bring this up because we need to modify our checklist once we have the bio nano or the nanobio, however we rephrase it. Once we have that narrow focus, we need to include those in those checklists. And that, once again, that helps our whole process vetting out what is our area that we want to go work on. These, for those of you not in the room, this is why it takes 2 years. There's multiple steps as it moves forward. And really, I don't see any major changes. One of the things we want to focus on, ISO, when we stray from this process, we end up taking 4 years. If we really follow the ISO process, which is used universally around the world, they have very defined steps on here's how you ballot, here's how you get comments, here's how you make your resolution changes, and going out for reballoting. So my advice is we stick to that actually proven ISO method. So on the next steps, really to the nanovio study group, I hand you my PowerPoint, at least my recommendations, and I'm sure there's much other input that you can find, but at least it's kind of a roadmap as we look forward. And hopefully, as a member on the Chairman's Advisory Group, we need you to come in and saying, we've given it a lot of thought, and here's our next steps as we go forward. And I'm seeing him stand up, so fortunately, I'm at the last slide, so the timing was good. But I do want to thank you. Standards, some people think they're somewhat boring to develop, but actually they're the most incredible. It's like a United Nations. I am so tickled to death. I look on this front page, or this front row, and you have the U.S. sitting next to Iran. And for very good reasons, because we come together and we work on standards development. So open it up to any questions.
Yeah, thanks for exact timing and nice presentation. We have time for one or two questions, so please raise your hand for questions. Okay, Dr. Lee?
To my understanding, your advice is to host the nanovire activity in current working groups. But I think that if it happens like that, then it may be there are something the problem like a working efficiency, because too many project or the items to discuss in one working group. So do you have any suggestion to overcome that kind of problem?
And if I understand your question, is there a different way that we can organize and the new work items coming in? We had extensive discussions before. Should we, just to organize and focus on the bio-nano, should we have a fifth working group? And I think the consensus was we're not at the stage. We don't even have a clear scope. And I think after we do these steps and we somewhat narrow the areas that we're going to work on, at that stage, we're always open. As a TC229, as the committee, we're always open to organize our work in the most efficient way method. But I do think we need to take these initial steps. So we even if we were to have a fifth working group today, how would we discern, hey, that is a metrology or that is a nomenclature? So I think as we move forward, and hopefully we can make some good progress between now and Edmonton, Canada, we can really set the stage of saying what is our long-term plan and how should we be organized. Clearly, if there was a nomenclature, we have an existing organization, even if we have a fifth or a sixth working group in the future, I think we'd still farm that out because that's where those expertise lie. But I think it's really going back into the study group of coming back and saying, these are the future directions that we're headed, and here's what our recommendations back to the Chairman's Advisory Group.
Thank you for the answer.
Yeah,
but
and
I would give you one more question about that.
Yeah, maybe we can discuss that during the coffee break. Yeah, maybe we are a little bit short of time. Any more questions?
I will certainly be here the rest of the day and tomorrow, so feel free at any time.
Okay, and thanks again for your nice presentation, and please give a round of applause to Steve. Our next speaker is Dr. Sunchul Hong from Bajitech Med. Actually, for this symposium, we try to gather people from the industry, standardization, as well as research. And Dr. Hong is from industry, but actually he used to be in academia. He used to be in Indiana University as a professor, and now he is a director in Bajitech Med, one of the nanovio company in Korea. Yeah, please give him a applause for his presentation. Thank you.
Okay, it's much less stressful talk for me instead of the science talk that I used to do. And thank you so much, representative for the ISO members for the, I can see the very hard work. And I realize that you guys doing all voluntary work. That is so nice of you guys, and thank you so much. And even visiting the nice country, the Korea, and thanks for that. And I am representing the company called BioTechMed in Chuncheon. And I'm very, very fortunate to join this company 2.5 years ago. And I'm very honored to talk about this company. And as well as the nanotechnology in medical diagnostics, which is, I'm not an expert. And we have an expert in Dr. Lee over there, and he provided nice slides that I can talk with. And thank you for that. And this is the company. Okay. Yeah, this is the company that I work with, and this is located in Chuncheon in Gangwon-do. And now we have 232 people working in this beautiful facility, and very, very family-friendly company with a nice kindergarten and playground for a lot of grown-ups as well. So nice. Okay, so this is the nice facilities around the mountain, and okay. So what the BioTechMed actually do, and we are a company working on the in vitro diagnostics company, and found in 1998. And we are basically using a so-called fluorescent lateral flow immunoassay technology, and basically using antibody-mediated. So I will explain that a little bit later. And the strength of this company is not only we making the reagent, we also making a diagnostic equipment for that. And since we've been struggling quite a long time, almost 10 years. Around 10 years ago, we made a breakthrough, and we means Dr. Che, founder, not me, actually. And he had made a nice breakthrough in around 2007 and 8. Since then, we never look back. It just grow up every year, twice and twice. And the last 5 years, our company grown to the 51% of the annual growth rate. And last year we made a revenue of $30 million. And we are expecting a lot more this year. And our customers are all around the world. So this is the yellow showing our customers located around the world. And basically, we are doing business with the 70 different countries around the world. We are very good at doing research and development. Actually, the founder of the company, Dr. Che, just was a professor until last year, and he quit school because it's too much work for him to do the teaching as well as teaching 200 people in the company. So now he's just doing only teaching 200 people in the company. We have around 13% of revenue on the R&D department. So last year we spent around $4 million for research, and we have 87 scientists, both in Korea and Qingdao in China, and the majority of them is actually located in Chuncheon area. And with a lot of PhDs, and majority of them is a master's. More than half of them is either PhD or master's degrees. What they do is actually they spend a lot of time doing research, and making many different reagents as well as the many different equipments as well. So we are actually have a lot of engineers and computer scientists and program and everybody practically. And we are sort of like to think that this company is mainly focused on the point of care diagnostics, which is actually part of the in vitro diagnostics, and using the so-called lateral flow immunoassay technology. I'm very good at this. So point of care technology, we try to get out of this business. What I mean is that not only in the point of care diagnostic, the point of care test, we try to the variety of the in vitro diagnostics and turn out the bigger company like Roche and all those companies actually now spending a lot more money and effort in the point of care technology. So we decide to spend more time as well. So actually, not only we are trying to expand it and realize, oh, this is actually very, very good business to doing a point of care technology. So we are actually spend a lot of effort on the so-called point of care technology. And actually, basically, point of care technology is when you go to see the doctor, you draw the blood and you send the sample to the so-called lab, clinical lab. Instead of doing that, you can just all the tests do it at the spot, at the doctor's office. You just, your nurses draw the blood, and then you can see, oh, you just you get the results within 10 minutes, 15 minutes, 20 minutes. Then by the time you see the doctor, all those numbers, your blood level, sugar level, and all those you have, HIV positive, negative, you can see that immediately. So you don't have to go through the three hours. Well, you come to visit the doctor's office next day. You don't have to do that. That kind of thing is actually much, much popular and economical, and that is called point of care technology. The problem in this business is actually sometimes we're doing it in so quickly at the spot. Sensitivity is the issue. Sometimes we have to sacrifice sensitivity. Second of all, sometimes each test, it could cost a little bit more than the lab test. That is the two major issues in the so-called point of care technologies. And nevertheless, this business is doing very well and continuously doing much better than last year. Last 5 years grows very fast, and this is showing the world in vitro diagnostic business market is like a $55 billion market, and point of care technology is cover up to almost 30%. So actually, this is become an even bigger pie in the in vitro diagnostic businesses. So we are actually very happy to see that. We didn't plan it; it just happened that way. It's just the world evolved this way. So we are very happy to see that. Okay, we are among the point of care, particularly very popular right now is the so-called infectious disease and molecular diagnostic. I cannot see it very good here. So those area is actually very, very popular, which requires higher sensitivity, which requires not traditional technology, but using a so-called much more sensitive techniques required. And this is our old-fashioned technology. It's very, very simple. Actually, I used to teach the immunology. I trained immunology, so I used to teach immunology and antibody and antibody-antigen interaction is quite basic, and went through so many Nobel Prize and everything. And this is how it works. When you're adding a sample here, sample means usually blood or urine sample, or it can be a sweat or all those urine sample, and you have all those sample interact with the antibody that reacts with a certain antigens. Let's say this guy's antigen, then you have the antibody react to it, then those sample with the antibody run through this direction by the way of the means of the absorption path, so-called wicking processes. So run through this way. When you have another antibody which is actually responding to this sample like this way, then you can get the nice signal here. And that basically this one is a control, internal control. So you have this kind of system. You can just, we've been using an antibody labeled with a fluorescence dye, which has been using for a long, long time, and we're still using it. And then you can see the fluorescence dye, reading the fluorescence by using the laser, and you can have a reader using the how strong, depending on the how strong signal is. That means that you have a lot of antigen is presenting in your sample, in your blood. Then you'll see, oh, you got some problem with that. You don't have it, you're lucky, something like that. That's how it works. So not only that reagent, we are making a variety of the readers, machines so-called, and tiny one, much better one. This is all you have to do is throw the sample blood in your tube, and then around 5 minutes later, you just have a waiting 5-minute start button, and then 5 minutes later, you got the printout saying that your number is this. So actually, fantastic machine. And we have a single one, multiple one, and this is actually measuring the concentration of hemoglobin in your blood. So actually, within a few seconds, you get those numbers. So these are the few of the readers we are developing, selling and developing. Actually, much more machine is in process right now. And not only that, we have a whole slew of the diagnostic the reagents available from the cardiac heart problem and the hormonal problem, including the pregnancy test. And you have the variety of the cancer treatment. Whether you have a cancer or not, you can see it in 15 to 20 minutes later. And with diabetes, HbA1c test, or all different. And also, we are very, very strong in the so-called inflammation and infectious agent, procalcitonin and C-reactive protein. And this is actually two of the very, very strong products we are doing very well. So not only all those equipment, actually all these reagents are made, and all the scientists keep making them one of the best products in the world, and that's why we are doing pretty well, I think. And using this very simple technique, we have so-called iChroma technology. We will get very high reproducible and broader detection range and accurate test results within a very short period of time. So that's what we are doing in our business. So we have a problem. We're doing pretty good. Can we do much better? Then what is the next step? So that is our issue. So could nanobiotechnology be the next commercial darling for an immunoassay in vitro diagnostics? That's the issue. So we hire many, many good scientists like Dr. Lee over there, and they are working on how to improve the sensitivity of our products. That's we spend a lot of time, and many of them is in progress, unfortunately. Some of them actually we are in the market pretty soon, end of the year, and so using the European, and they'll also explain to you a little bit. And basically, I can see the two different way of the nanoparticle in lateral flow immunoassay. One of them is basically increase the sensitivity, and second one is concentrating the sample. Basically, you're using a nanoparticle with a much sensitive dye, such as lanthanum group, and europium is one example, or quantum dot is another example. You can get a much higher sensitivity. And second part is that you concentrate the sample. I don't want to say that, but actually once you have the diarrhea and you want to know what kind of bug you have. So basically, it's all diluted. And you can actually concentrate the dose sample using the what? Magnet. So magnet is a wonderful tool to concentrate the sample. Or you can have in your blood certain samples that, such as you have the MI, high tech, and you have a certain protein is floating around in your blood, and very, very low concentration. You can concentrate that, and you can actually detect a very, very small amount of the protein. You can catch it. That can be done all by nanoparticle, using magnetic nanoparticle. That is a two major use of nanotechnology in diagnostic businesses. Am I okay? Okay, so I'll be quick. And so using a high sensitivity, we are using a europium and quantum dot. Basically, we are using a quantum dots, and the magnetic use of magnetic nanoparticle for the purpose of concentrating it. We are doing basically magnets actually. This is the use of the nanoparticle. Instead of just the dye, usually dye means fluorescence dye directly conjugates the antibody. That's what we've been using for a long time. Instead of doing that, we can actually adding a very strong dye without no background material, and those things you can actually put it into nanoparticle. Then you can adding an antibody on surface of the nanoparticle. You can have a specificity as well as the much more sensitive detection of the samples. So that's what we are using it. And so this is actually you can compare to the fluorescence stuff, which is actually much higher background. When you're using a nanoparticle, you have a clean-cut background. So this is the use of the europium particle. You can have a much lower background. Since our system does not, people might know the ELISA. ELISA has an extensive wash, but actually all our system does not have any washing step. Just adding a blood, run through it, within 5-10 minutes later you get the results. Because of that much background problem can be recovered by using the nanoparticle without much cleaner background. This is the case of using a europium, and we've been using a europium, and we tried using a terbium. The reason I wasn't sure why we are not using it, and the Dr. Lee told me that it's very expensive, so it's not commercially usable to use it. So that's why we are using it. Not only that, since they are using collecting all those signal for a long period of time, so-called time-reserved emission, so we've been using that technology as well. So for the concentration, this is one of the example of the concentrating the samples, and you can get rid of, this is a red blood cell. You can get rid of all the red blood cell from the red blood, whole blood, using the magnet, and this is using a magnet, and then all those red blood cell can be cleared out using the magnetic beads. Magnetic beads, with all the magnetic bead covered with antibody that directly binds the red blood cell, then all the red blood cell is bound to the antibody, and you can fishing out all the magnet, using the magnet, all the red blood cell here. And actually, since I'm immunologist, my lab, when I was running the lab, I've been using a magnetic bead all the time without knowing that I'm using a nanoparticle. So actually, I've been using it for no more than 20 years, purifying the T cell, purifying the stem cell, and all sorts of things. And now I realize that has been used, magnetic nanoparticle covered with the antibody. So I've been using it, Dynabeads and all those company things have been using it for a long time. And actually commercially, so Mitsubishi company called Mitsubishi company make the machine called the PathFast, which is using a magnetic bead technology, and they are using it for the commercial use to measure the so-called troponin I, which is actually nice biomarker for heart attack. So this is the PathFast, and this is basically using a magnet nanoparticle. You can concentrate in the samples, and I've been using those things a lot. Okay, so I think I said enough, so I will stop it. Thank you very much for your attention.
Yeah, thanks for very insightful talks, and he's also very exact in time. But let's have just one question, if there is. Raise your hands if you, yeah, please.
I was just curious, on your diagnostic, on the one in commercial use, can you have multiple compounds that you're testing for, or is it each one is specific to a specific compound you're looking for?
But actually we're doing one at a time because of the economical purposes. You can do many different things at the same time. One strip you can have a multiple line, you can do it, but actually sensitivity, cross reactivity, all sorts of things at the same time, it's much economical for our company's point of view, just as making one at a time.
Right. Thank you.
And thanks again for your nice presentation, and please give a warm applause. Thank you. Okay, so it's time for a coffee break. So we have about 20 minutes, and thanks for all the speakers with the exact timing. So we can use all 20 minutes, and we will reconvene in 3:30 and here. Okay, thanks, and enjoy the coffee break outside. also coming back in August for IUPAC.
Yes, yes, in
Yeah.
Busan. Will you be going? to
Actually, I'm also convener for the one of the session, nanosafe session. But I saw your name in Nanovio.
Busan? Yes, yes.
Okay,
Bye-bye.
great.
Okay.
So have you been to Korea before?
Yes,
Ah.
quite a few times.
Yeah. So I was a little bit worried about like because if you first time, then you may be a little bit difficult to.
No, no, it's fine. It's just that Seoul has become so big and
Yeah.
crowded.
Oh, yeah.
The traffic is very.
But if you can take the
Busy.
subways or
Yes, yes, yes.
then it will be convenient. And if you have any assistant
Yeah.
for while you are staying, just ask me.
Okay.
Yeah,
Sure, no
I'm
problem.
just living nearby, so
Okay.
I can give you some guide.
Okay, no
Yeah,
problem.
yeah.
We are fine.
네. 아, 예, 여기서 이쪽 뒤에 있는 걸로 하시면 될 거예요. 지금 이거는 제키인 거고. 오케이, 두 번째 세션 시작하겠습니다. 이제 들어와 주세요. So please come in, and we will begin the second session very soon. Yes, she is very comfortable. So you studied in Princeton, Princeton and was at MIT and returned to Singapore for the current it's institute or research institute or university.
Start this new institute.
Ah, okay.
It's not university.
Okay, so I will maybe introduce just briefly so that we can have more time for presentation and stuff. Okay. Maybe you can just yeah, sit on there. 예, 이제 들어와 주십시오. Please come in. We will begin the second session. Read in a minute. Yeah, thanks for returning to the second session of the Nanovio Symposium. And the first speaker for the second session is Dr. Jackie Ying from Singapore. She is very famous researcher, and she did his PhD in Princeton and worked for at MIT for several years, and now he returned to Singapore and have the Institute of Bioengineering and Nanotechnology. And she will present about the synthesis and applications of nanostructured biomaterials and biosystems. And please give him a warm applause for his presentation. Thank you.
Thank you for the kind introduction. It's a great pleasure to be back in Seoul. It's really one of our favorite cities. Today I would like to speak on the synthesis and applications of nanostructured biomaterials and biosystems. And here in my research group, in this research institute that I founded about 12 years ago, we are focusing on the synthesis and functionalization of nanostructures. In particular, we're looking at how not only we can control the material's particle size by introducing dopant, coating, and other nanoparticles so that we can really functionalize this material for various applications that ranges from pharmaceutical synthesis, green chemistry and energy.
compound toxicity assessment. Are you almost ready?
Yes.
Okay. Maybe the screen is not ready yet. Can you change the screen with a different computer? 이거 연결해 주시는 분이 Yeah, although we are not intended to have this program for these kinds of smooth connections, but looks like those nanoparticles and lateral flow and diagnostics and Professor Jackie's talk, they're all well connected. And I think there are a lot of space for nanobio standardizations. Okay, ready? And I think most of you may recognize it. So there are a couple of cameras, and actually this symposium is lively streamed on YouTube. So if you search on YouTube with Nanobio, then you may see your face. Okay, looks like it's ready. And yeah, please give him a loud applause for his presentation. Thank you.
Thank you, Professor Yun, for the kind introductions. And I would like to appreciate giving me a chance to introduce my research in this wonderful MBS 2015. For laypersons who has a different background from me, I'd like to basically give you the basic concepts of the microwave bioprinting technique we are using for assessing the compound toxicities. As Steve mentioned, soju is different from water. So this is actually one of my favorite slides. I'd like to start off with these questions. Have you ever wondered why your friends or colleagues can drink more than you can drink or less than you can drink? If your answer is no, then this slide gives you some idea what happens actually in the liver. Ethanol in the alcoholic beverage is converted to acetaldehyde by this alcohol dehydrogenase, and the enzyme called the cytochrome P450-2E1s. And then this aldehyde, acetaldehyde, can be further transformed into acetate, and this acetate can be completely degraded into carbon dioxide and water. When you have accumulations of this acetaldehyde in your liver, it can create a couple of side effects. It can cause a headache. It can turn your face colors in red. And then, as you notice here, from the reactions of cytochrome P450s, it can generate the compound called reactive oxygen species that can change the structure of DNAs, eventually leading to hepatotoxicities. So that's why people said that, oh, heavy drinking is really bad for your health. Okay, and one of the things I want to point out is that the cytochrome P450-2E1 can be induced by ethanol, which means that if you drink every day, this enzyme can be induced more so that you can drink a lot more than what you used to drink. So what happens is that each individual has different levels of these enzymes in the liver. So let's assume that if you have a less amount of this enzyme called ALDH in your liver, then you have a lot of acetaldehyde accumulated in the liver. And you cannot really tolerate heavy drinking. So I'm one of these people who cannot tolerate the alcohol consumptions. The second thing is, same thing can really happens when you're taking the drugs. So this is another slide giving you some idea about the drug metabolism. You may have heard about the acetaminophen, which is the active ingredient of Tylenols. And this acetaminophen can be transformed into several different intermediates by a couple of enzymes called the drug metabolizing enzymes, such as glutathione transferase, again, cytochrome P450s, and the sulfur transferase. And among these intermediates, these quinoforms are toxic to the liver by forming protein adduct and then causing oxidative stress. And all of these enzyme levels can be buried in different segments of populations. So knowing how much of these enzymes expressed in the liver is very, very important in terms of the drug metabolism and related toxicities. And what we're trying to do is the developing some kind of assay tools to predict the adverse drug responses, especially in the individual levels. So to better explain what really happens, I'd like to explain a little bit about the metabolisms and the phenomena happens when you're taking the drugs. So drugs are basically, I'm just talking about the chemical drugs, the oral drugs. Drugs are basically absorbed through intestines and goes directly to the liver where the metabolism occurs. And then drug and its metabolite can be distributed to the entire bodies, organs, and eventually eliminated through kidneys. And in the liver, drugs are often converted by drug metabolizing enzymes such as cytochrome P450 again. And these drugs and drug metabolite can inhibit activity of these drug metabolizing enzymes, which create complicated situations like metabolic stabilities and drug-drug interaction problems. So even bigger problem is that each individual has the different levels of drug metabolizing enzymes. And along with these genetic polymorphisms, making the assessment of these drug toxicities and the metabolism is very difficult. And here is the couple of examples of drug metabolizing enzymes, which is basically a group of oxidative and conjugative enzymes. And the oxidative enzyme is typically called phase 1 enzymes, and it can catalyze oxidation reactions. And these oxidation reactions often generate the reactive oxygen species or the reactive drug metabolites. And then the conjugative enzymes include including the UGT, GST, glutathione acid transferase, or the sulfur transferase. These are the enzymes increase the solubility of the drugs by adding bulky hydrophilic groups to the hydrophobic drugs. So it can eventually enhance the eliminations of the drugs from the bodies. As I mentioned earlier, the drug metabolizing enzymes such as cytochrome P450 is not just the single enzymes. It has a lot of different isoforms within the group of the cytochrome P450 families. And the genetic gene expression levels really varies dramatically, sometimes over 1,000-fold, which create complicated idiosyncratic or the independent adverse drug responses. So assuming that you have 1,000-fold different expression levels of these enzymes in the bodies, then your drug response, metabolism, and related toxicity can be also varied 1,000-fold. So assessing genetic variations of this drug metabolism in the bodies are one of the really important area of research. So one way to address this problem is to using the biochip platforms. Basically, biochip platforms are classified into two categories, one of which is microarray biochips, and then the other one is microfluidic biochips. Microarray biochips refers to glass, silicons, or plastic chips with the DNA, proteins, virus, cells, and sometimes tissues. If you have a DNA microarrays on the silicon wafers, it is typically called DNA chips. One of the most famous example is the gene chip from AP Matrix. And as you know, the microfluidic biochips refers to the chip with the microchannels, mostly for the diagnostic purposes. It's used for the diagnostic purposes. So my research goal at Cleveland State University is to develop innovative microarray biochip platforms and miniaturize biochemical and cell-based assays on these micropillar and microwell chip platforms. And this platform is mainly used for the preclinical in vitro evaluations of the drug toxicity and efficacies. And we use two main instruments, which is developed in collaboration with Samsung, one of which is called microarray spotters, and the other one is called the chip scanners. The microarray spotter is nothing but the biological sample printers. And then the chip scanner is an automated fluorescent microscope. The movie is not working, so let me just turn on this movie. So this movie shows how the machine works. In terms of the principles of printing biological samples on the chip platforms, it is very similar to the inkjet printers. Instead of printing inks on the papers, we print the biological samples on the chip platforms. It can aspirate the samples from the 96 well plates like this, and then measure the droplet volumes using the camera on the side here, and then finally dispense the accurate amount of the samples on the chip platforms. And everything is automated, so you don't need to touch anything once it's started, and it's computer controlled. And let me introduce this micropillar chips and explain how it works. Micropillar chip is the plastic chips with 532 micropillars onto which 3D cells are growing. The cells, cell droplet as small as about 40 nanoliters can be dispensed using these microarray spotters onto chip platforms. And then cell spots are strongly attached on the chip surfaces through the surface chemistry we developed. These chips are basically aminely active surfaces, so it can covalently attach the proteins to provide the high affinity to these to the hydrogels. We tested hydrogels such as alginate, matrigels, pure matrix, and fibrins, and we are also studying more biocompatibles and the tunable hydrogels in collaboration with a couple of professors in the chemistry. And this is the scanned image of the chip with the human liver cells. As you can see, the cells are growing in tiny 40 nanoliter spots and forming 3D structures over time. And the cell spot volumes are as small as about 40 nanoliters, which requires extremely small sample volumes for biological assays. And this is particularly beneficial for the primary human cells or the cancer cells isolated from the patients. And this slide illustrates the couple of examples of the cells encapsulated on the chip platform. And as I show you here, we were able to encapsulate the primary human cells and transformed cell lines, as well as the red primary and red transformed cell lines. And we were also able to encapsulate the stem cells also on the chip platform. This is micro well chips with 532 micro wells. It can accommodate the recombinant adenoviruses. The sample volume here is about 800 nanoliters, and it is complementary to the micropillar chips. And instead of, in addition to these recombinant adenoviruses, we can print the compounds, proteins, reagent also into these microwells. To mimic to simulate these metabolism-induced toxicities, we constructed recombinant adenoviruses carrying genes for drug metabolizing enzymes. This is a couple of examples of the recombinant adenoviruses carrying cytochrome P450 genes, UGT genes, or SIRT genes, or the GST genes. So by simply sandwiching these chips two chips together, we were able to expose cells to the recombinant viruses so that cells can express the drug metabolizing enzymes. This stamping assistant apparatus and also the chip structures allow us to accurately sandwich two chips together. And once the chips are sandwiched, then we can incubate those cells, the chips, over a period of time, such as about 3 days, without any evaporation issues. And this will allow us to grow the cells uniformly on the chip platforms. And once the cell culture and testing with the compound is completed, then you can stain these chips with the fluorescent dyes. And using this the staining plate, so entire chips are just stained with the staining solutions. And after the staining, we were able to detect the different property of the property or the features of the 3D culture cells on the chip platform. Here I give you a couple of examples of the cell images stained with different fluorescent dyes. So for example, if you want to know the integrity of the nucleuses, we can use Horchester dyes. And also, we can use the TMRM to measure the health of the mitochondrial membranes. So this is really the high content imaging assays on the chip platforms. And the scale is, again, extremely small compared to the conventional 96 well plate assays based on the monolayers of cells. And once you obtain the images, the next step is data analysis. We were able to analyze the images using the in-house the software, and also we can use the ImageJ, this open source free software to analyze the cell images. It basically extracts the fluorescent intensity from the images and then plot the dose response curves and calculate the IC50 values. So it goes into more details about this, the metabolism-induced drug toxicities. Let me talk a little bit about the fundamental questions I had. So what I wanted to do is simulating what happens in the liver, especially in vivos. And to do that, I need to come up with a good way to infect the cells. And we have tested different the gene infection, transduction and transfection method using lipopectamines or the viral delivery systems. And then we wanted to precisely control the expression levels of these drug metabolizing enzymes. So we have tested it again with the adenoviruses with the different concentrations of these viruses. And we need to know whether this enzyme is still active, right? If the enzyme is denatured within the cells, then there is no way you can measure the drug adverse drug responses. And the scale is so small, so we need to make sure that scale down doesn't affect the cell viability or doesn't change the property of the cells. And there are about 7 different, as far as I know, the human liver cells available. So we need to know also which cells actually works best among those human liver cells. And the most important thing is whether or not we can mimic the adverse drug reactions in vivo. And finally, as you know, these drug expressions of the drug metabolizing enzymes vary dramatically in individuals. So we need to somehow, we need to know how we can control the 100 or thousands of different combinations of drug, the combinations of metabolizing enzyme expressions on the chip platform. So I'm going to briefly introduce the basic concepts and then how we demonstrate these concepts on the chip platforms. So this is optimized the protocol for the chip experiment. First, cells are printed on the micropillar chips and then sandwiched onto the microwell chip containing the growth media and cultured for about 1 day to maintain high cell viability. And then these cells, the pillar chips, are detached from the microwell chip containing the growth media and then sandwiched onto the microwell chip, another microwell chip containing the recombinant adenoviruses. And after 1 day of culturing, we discarded the virus chips, and then the micropillar chips expressing now the drug metabolizing enzymes in different spots, incubated with the compounds in another microwell chips. And after about 2 days of the incubation with compounds, we now stain these cells with the fluorescent dyes to detect the live and dead cells. And after the staining, we scan the chips and then analyze the images of the cells. Here's one example that show us how we can control the expression levels of green fluorescent proteins, which is called GFPs, and the red fluorescent proteins, which is RFP. And by simply changing the concentrations of these, the adenoviruses carrying genes for GFPs and the genes for RFP, we were able to control the amount of the protein expressed within the cells. So this is really the proof of concept studies to demonstrate we can control the expression levels of the proteins within the cells. And then, with this encouraging result, we wanted to demonstrate that we can express different combinations of drug metabolizing enzymes. So we selected 3 representative drug metabolizing enzymes, such as cytochrome P453A4, CYP2C9, and UGT1A4. And we prepared 3 different mixtures with the maximum of 15 MOI. MOI is basically the multiplicity of infections, which is basically the concentrations of the virus solutions compared to the number of cells. So using this randomly selected 3 different mixtures, we ran the Western blot analysis to know how much of the proteins are actually expressed within the cells. And then, as you can see from this graph, we were able to control the expression levels of these individual, actually the mixture of the 3 enzymes, by simply changing the concentration of virus, the recombinant adenoviruses. And this was done in the 6-well plate with the 2D grown cells, and we wanted to demonstrate the same concepts with the cells grown on the 3D, the chip platforms. So we used the in-cell immunofluorescent assays to measure the expression levels of these, the drug metabolizing enzymes on the chip platforms. So this is the scanned image of the chips with the different levels of drug metabolizing enzymes on the chip platforms. So again, we used basically the same set that I just mentioned earlier with the 2D cells, and we were able to control the expression levels of drug metabolizing enzyme by simply changing the concentration of the adenoviruses. So one thing I want to point out is that the expression levels are almost identical to the expression levels of the drug metabolizing enzymes in the 2D grown cells. And with this encouraging result, we wanted to simulate the metabolic pathways of the tamoxifen, which is the estrogen receptor positive drugs for the estrogen receptor positive breast cancers. And as you can see from these schematics, it's really busy schematics, right? There are lots of enzymes involved in the metabolisms of this tamoxifen. And oftentimes, these parent compounds can be very toxic in the liver. And the metabolite of this tamoxifen is actually more potent than the parent compound. So depending on the expression levels of these enzymes in the liver, their toxicity and their efficacy can be changed. So using 100 different combinations of these adenovirus on the chip platforms, we wanted to, 1 minute. So we demonstrate that we were able to construct 144 different combinations of the viruses on a single chips by printing the virus in a combinatorial fashion. And then we were able to identify the hits with the different combination of enzymes. So this hit can provide the metabolic enzyme condition that allow the cells to survive under a high concentration of tamoxifen. So let me just summarize the microarray biochip platforms. It is relevant for the mechanistic toxicity assays, and it requires extremely small sample volumes. And this method can be expanded to other cell types to measure the organ toxicities. And then we can control the expression levels of drug metabolizing enzyme to simulate the individual dependent drug metabolisms. And finally, it can be extended to other cell-based assays, and especially the gain or loss of function studies. Thank you. I'm more than happy to answer your questions.
And thanks for nice talk. And we can have just one question for him. Yeah, please raise your hand if you have any questions. Yeah, maybe. Yeah, since we have a little bit like behind the time, so maybe we can give him a loud applause for his presentation. Thank you very much. Yeah, we will go back to the standardization issue. The next speaker is Professor Ali Betolai from the Iran University of Science and Technology. and he will present about the nano standardization and nano safety program and activities in Iran. Yeah. Are you almost ready?
Yeah.
Okay.
Thank you. First of all, I would like to thank the organizer for inviting me and colleagues to this very important meeting. I think Korea had taken a very good step during this event and the last 2 days regarding the interim meeting. We had very constructive meeting, and thanks to Korea for hosting this very important event. I think this will be a good lesson for other countries also to engage more in terms of providing the infrastructure and also the chance for further progress of the projects. Actually, my talk will be a rather different to what has been actually delivered. Very nice lectures that has been delivered by colleagues. Actually, I'm a university professor in material science in Iran University of Science and Technology. And meanwhile, I act as the director of nanostandardization and international collaboration of INIC. I thought it might be a good idea to briefly actually introduce INIC. Excuse me, I don't know whether. So very briefly, I will introduce about INIC and talk about it and some of the achievements that the country have had. But mainly I will focus on nanostandardization and nanosafety activities and achievements that we have had. And also I will speak about the nanosafety network that we set up a couple of years ago in order to consider these very important issues. And also one important scheme that we have set up called as NanoMark and what we will do in future. So the INIC is affiliated with the Vice Presidency for Science and Technology. It is in charge of policy making, public awareness, supporting nanotechnology development in the country, and also meanwhile it considers the evaluation and assessment of the activities and the achievements in the country. Very brief background and history of the INIC. We set up the nanotechnology policy study in 2001, and in 2003 officially we set up the INIC, and formally we had our national initiative program passed by the cabinet at 2005. This is the current structure of INIC, which looks after nearly everything in the country, from training kids at school up to commercialization through the various sectors that has been actually set up for doing so. So this is just a very brief description, and I would say that 6 ministers act as our board of trustees, and different sectors, as you see, are involved in our program. Anyway, our national program, the first 10 years is now about to be finished, and we are in the process of developing our second 10-year program, which will cover up to 2025. The vision of program is creation of wealth and raising people's quality of life. We have managed to, during the last 10 years, to develop nanotechnology in a quite good level in terms of especially human resources that we have trained. Now 37 universities are providing MSc programs and 17 PhD. Amongst them, Nanovio, a couple of universities are providing the curriculums related to nanobio. Well, we have supported various researchers. Now we have a mass of more than 2,000 faculty members who are involved in nanotech research. And above MSc, we can say that more than 20,000 experts are active or have been active in this field. a rather quantum jump in terms of the internationally peer-reviewed publications from the time we started in 2001, and in 2014 we could actually publish more than 5,400 papers. This shows the number of ISI articles that has been published. I mean, worldwide. You see, Iran is doing quite good, but we are still in the early days. And in terms of the quality of the papers, it's quite good, but it needs, of course, much more improvement. But anyway, this shows our willingness towards science generation in the field of nano. We have set up an infrastructure to provide the necessary services to scientists, researchers, and also to our industry by setting up nanotechnology lab network, it is called. They share more than 1,000 equipment between 70 research centers throughout Iran. They provide various services. And the slide here shows a number of services that have been provided to the outside customers, i.e. either from industry or from some other universities and the rest of it. And they could earn quite good amount of money anyway through this such. In terms of the startup companies, more than 100 companies in different areas. I would not go through them. Manufacturing companies, more than 150, again, in different areas, including healthcare and the others. Also, more than 270 products has already been introduced into the market by our companies. I just want to emphasize in one of them, we have a special program on nanomedicine. Up to now, 4 nanomedicine actually has been introduced in our market. One of the most important one is this sinadoxazome, is an anti-cancer liposomal drug. US is another country who produced this, and others in some European countries, they produce under the license that US have released, but this is based on our own international patents. And it is 2 years in the market. And also there are another 3 nanomedicine. Okay, if I come to, after this very short and brief introduction of nano activity in Iran, if I come back to the main issue, that is standardization, I will explain you a short story that why we engaged in nano standardization. Actually, it was in India that I was speaking about Iran's activity in nanotechnology. After my talk, a guy came to me and said that, okay, if Iran has a national initiative program, why you don't engage in nano standardization? And that time I didn't know that this program has been initiated, and the guy was Peter Hatto, that you all know, a very nice guy, that he actually invited us to engage into this program. Anyway, after we participated in the first event, when I went back home, I set up the mirror committee to ISO TC229 back home with 3 working groups. And I hope that in near future, we would also introduce Nanovio working group in Iran as well, just to actually share in this very important actually activity to be actually materialized hopefully in near future in TC229. Anyway, these are experts from universities, various industries, or standardization actually institute. This is the website that people can have access to that. Anyway, up to now we have published more than 30 national standards in the field of nanosafety, environment, measurement, and terminology. And as you may know, Iran has led 3 ISO standards, and we want to do much more than before. We have established a very important committee in our Ministry of Health. We call it Iran Nano FDA, which deals with the development of the protocols necessary and related to the nanoproducts which have some issues related to EHS. So products containing cosmetics, pharmaceutical, healthcare, and medical devices which are in the field of nanotechnology need to get certificates and allowance from this committee to go to the market. And even recently we have approached our customs to actually, because the goods are sometimes legally, sometimes illegally are imported into the country, and we have asked also customs to act on it, and so they look also into the imported goods as well. We have also established a standard committee in Ministry of Agriculture. You know, nano has got a wide range of applications in agriculture. We have designed a comprehensive database for nanotechnology standards and safety. And also we support master and PhD theses related to nanotech standardization and safety issues. Well, in terms of the public awareness and also such areas, we have been holding various workshops and seminars, both nationally and internationally. In 2015, we had quite good activity in this regard. We had an International Nanosafety Congress, and also NFC. This was a big conference in Kish Island, one part of which was devoted to nanosafety and other activities. Well, another a step that we took was setting up this Iran Nanosafety Network, which actually aims are as the following: for sure, networking researchers and experts and institutes and industrial units active in the field of EHS, to focus on national priorities in this area, and also to come up with the draft of national or international standards, again related to EHS issue. This slide shows the structure of our nanosafety network. Meanwhile, we are a member of Asian Nanosafety Network. So this slide shows the structure of the network. You see we have 4 groups: occupational safety working group, human health working group, environmental health and also nanoethics. These are the 4 groups that we have set up, and they have their own scope and focus of area. For example, in terms of environmental health, the scope of this WG includes drafting standards, regulations, and guidelines for environment and agriculture in the field of nanotech. These are I don't want to go through all of these, the main areas of focus in relevant areas. For the occupational safety, this includes human resources, training, drafting the relevant standards and protocols. Again, their main area of focus: assessing processes and factors affecting the level of exposure, identifying the groups who can be exposed to nanomaterial, studying the effect of exposure with different types of nanomaterial, and the rest of it. By the way, we have developed also a software for students and even for kids at high schools, because they are now trained in Iran about nanotechnology regarding the nanosafety issues as well. Also for human health, again, these are the areas of focus: in vivo effects of nanomaterials in human, characterization, effect of physicochemical parameters on toxicity, and the rest of it. Due to the shortage of time, I would not go through this. And nanoethics, this is something that it is less touched, to be honest, in the world. And by considering this issue, we have set up this committee. You know that we have the bioethics regulations, but not the nanoethics. I don't know, maybe in future in ISO we may also involve in that. Anyway, but this is the committee that we have set up for actually devising the guidelines, regulations, codes of conducts, and related laws to support sustainable and responsible development of nanotech. Another important step that we have taken, and Iran is among the 3 countries who have come up with a scheme to support consumer trust and confidence, is the nanomark scheme. Taiwan is the first country who set up this scheme. Iran was the second, and the third one is Thailand. And some other Asian countries also are considering, as far as I know, about such a scheme. This would help to boost market development and facilitate marketing and trading. A lot of advantages, actually, mainly for supporting sustainable growth of nanomarket and preventing frauds and misleading. This is an important issue that this actually scheme follows. The nanomarket scheme in Iran considers all these 4 aspects, the being nanoscale or utilizing nanotechnology, safety issue, labeling and efficacy and durability of the actually product. This is the structure that we have set up in Iran, which actually invites all of the major players in order to provide this mark to the product. So our Institute of Standards, Iran Nanosafety Network, representatives from Ministry of Agriculture, Ministry of Health, and also the customs. So this is an important step that also will be soon implemented in nationwide level. We expect after 3 years, up to 5 years, it will be mandatory. Every nano product that are produced in Iran would have this nanomark, and every goods that are going to be imported into Iran is going to comply with the requirement of nanomark. As the last slide, some of the future programs that we plan. In our next 10-year plan, we have targeted to develop more than 200 national standards. We would like to increase our contribution to ISO TC 229. As I mentioned, we will implement nanomark scheme mandatory in future. We would like to reinforce our regional and international collaboration for developing nano standards and issues related to nano safety. And we will support the development and commercialization of reference material. We have a program for reference material. It is in the early stage, but we think that we need to develop some of the reference materials. And also we will have a big program that is the development of our National Nanometrology Center. Currently, we have developed our National Nanometrology Initiative that actually considers all of the steps that the country needs to take. Sorry for this very fast and quick presentation. Just I wanted to share some of our activities, and thank you for your attention.
Yeah, thanks for very informative talks, Professor Ali. And we have one more speakers today, and our next speaker. Oh, sorry, I forgot to ask questions. And sorry. So if there is any questions, we have a time for just one quick questions. Yeah, please.
How many products have you considered the nanomark for?
Actually, since this scheme is new, actually currently it is voluntarily. Those companies who want to take advantage of our incentives, they should definitely get those nanomark. Because we should provide some time to companies in order to get familiar with the scheme and the rules and the regulations, and then it will be mandatory for all of the products. But currently it is voluntary. Every company, because we also provide incentives to companies to, our budget is for such reasons. So in order they take advantage of these incentives, they should comply with our rules. So And I imagine in the next year, at least it will be more than 2025, something like that.
Okay.
Yeah.
Thank you.
And any other questions? Ah, yes, just one more.
Ali, thank you for your talk. And as I know, Iranian is investing a lot of money to the nanobio. And also you have a lot of universities and institutions researching about nanobio. Do you have any governmental plan for the long-term plan for the nanobio?
Actually, one of the reasons that I took part here, and I was also supporting our colleagues remember from the previous ISO meeting, it was due to such interest that is available in the country. And because we have, especially on nanomedicine, drug delivery, targeted drug delivery, and also diagnosis, we have a actually focus program on that. And because of that, for sure, we do seek the collaboration with other nations and also through the ISO. This is very important. And yes, we are very keen and we will be very happy, really. And also, I would say that we have taken some steps. I hope that once the committee is set up, we will come up with a basket of some new work item proposals based on the previous works that has been done in my country.
And thank you very much. Yeah, our last speaker is Chang-Ha Lee from Mecasys, and he will talk about the examples of analytical instruments using nanobiotechnology and the effectiveness of the standardization. And before his talk begin, there is one announcement. So after this talk and after we do some panel discussion, the dinner session is ready in the next room across the hall. And I think we have enough seat available. So everyone in this room, including all the like general audience, are welcome to join for the dinner session. Are you ready? Okay. Thank you.
Nice to meet you. I'm Chang-Ha Lee. First of all, I would like to thank Professor Yoon and all the member of the TC229 for having me here and then giving me a chance to introduce our company. I think our company is rather far from these discussions, but it will be really appreciated that if you can understand the
Okay, we 이제 거의 끝나가지고. So we have only one last like panel discussions. Actually, I will pass the microphone to Dr. Taegol Lee from Chris, and he will introduce a little bit about the issues in the panel discussion, and he will proceed for further discussions. Yeah. Thank you very much.
Thank you, Chairman. So it is wonderful, fantastic the symposium so far. So my name is Tae Lee from Chris. So today we're gonna have a panel discussion for like 30 minutes or 20 minutes, if you wish, if you're hungry, and then we can go to the next room. So I'm gonna briefly summarize our activity, Nanobio or NBS, a study group in ISO TC 229. And then we're gonna talk about a little bit which area we should standardize first, or which area we need standardization for the industry, because the looks like we have a consensus. We need to make a good standardization for stakeholders, industry needs. So, but we don't want to do the duplication from the existing standardization. So as Simon mentioned, our the structure here, ISO TC 229. So we have 4 working group, and then 2 test group, and 1 study group. So we are working very, very hard to make a good standardization. I used to be in the ISO TC 201. So we have like 10 people in every meeting. So, and then we are making the 1 or 2 standardization in year. But here is we are making so many good standards so far. So we want to keep working hard on making the good standard in nanotechnology. So the 2 years ago in Mexico, we have a 2 years ago, we had a very long discussion about we need to have some kind of study group related to the nanobio. So then, at that time, we have a kind of consensus. Nanotechnology converged to the biotechnology, and also we had OECD workshop to deal with we need to have some protocol to measure the nanoparticle and inside different environment. And then we did a survey on nanobio areas, and then the ISO standardization requirement, and then in Brazil meeting, we had a very good long discussion about our study group name, Nanobio. So we changed our name from Nanobio to the nanotechnology and biological system. But I'm not sure this name is also correct for our purpose. So we keep working on our name. And then also we established the mission and scope and objectives of our study group, NBS. And then also we work very hard to establish the interested area related to the nanobio. And then we circulate. And then last year in India, New Delhi, we decided to start standardization related to nanobio in existing working group. And then Steve mentioned, and then we just go for it, and then we'll see what happens in existing working group. So we're gonna still keep going, keep working on the nanobio areas, and then we'll try to find how we can accommodate our activity in ISO TC 229, also ISO TC 276. And then here, our members, like in the Nanobio study group, we got a nomination from the a member body, every country's member body, and then we have 16 countries, and then 4 organizations, and then we have 71 experts. It's quite big experts so far. And then this one is the established area related to the nanobio. And the nomenclature is related to working group 1. So it's and then nanomaterial characterization in biological systems, working group 2, related to working group 2. And then EHS, environment, health, and safety, related to working group 3. And then material specifications, related to the working group 4. And then we have also other 2 areas, and then diagnostics and imaging, and then therapeutics. So we heard today many on diagnostics, in vitro diagnostics, and then imaging, and therapeutic nanomedicine, or nanobio sensors, and high throughput screening. And then this all kind of is related to like kind of systems and products. So we are not sure this kind of area is how can overlap with the existing working group. So it's kind of a question, nanobio, what's the scope of nanobio? So it's kind of temporary, the development standard, which are interest in industry for nanobiotechnology. Nanobiotechnology means application of nanoscience or nanotechnology to biotechnology or biology. This one is just temporary on current at this moment, but it can be changed. And then also we heard the many concerns from the regulatory officer, regulatory institute. So we put some note here. Exclude nanobio standard, which are currently covered by existing regulatory agencies, but supported as needed. So you want to support the regulatory of the agency, not intervene or something. And then I'm going to briefly show you about our survey result. And then bioimaging contrast agent is very important in our nanobio area. So 37% is related to magnetic resonance imaging nanomaterial. As the Professor Hyun pointed out, is very important in the MRI. And also 20% here related to the enhanced source nanoparticle, and also fluorescence nanomaterial too. And then many people are talking about the safety and the function evaluation method related to the contrast agent. It should be standardized. And also terminology, and also development of standards for the contrast agent, etc. And also, we heard about nanomedicine, drug delivery, diagnosis, and then also these are very important area and treatment. And also, we concern about biocompatibility standard, toxicity, and then standards need to be in the drug delivery standard and the efficiency, drug loading efficiency, and the tumor targeting standard, tumor targeting efficiency, etc. And then the third one is the tissue regeneration stem cell. And then these days, stem cell is very important for the medical application. So it's wound healing and then controlling. And then many people, they are trying to use the nanoparticle nanomaterial to the effect on the stem cells. So nanoparticle safety to stem cell, regeneration factor from stem cell, and the terminology and the nomenclature. And then also the in vitro diagnostic. The last area is the nanobio sensors and diagnosis. And then the Professor Lee talking about the high throughput MEMS and then NEMS, and the high throughput the assay microfluidic device. And then this one is very important in also in vitro diagnosis. And then also DNA chip and then protein chip. Also they want to use the nanomaterial to increase the sensitivity and specificity. So these areas should be standardized. cellular condition for in vitro nanobiosensors, and the physical chemical property of nanoparticle for nanobiosensors, and then protocol for measuring fluorescence or electrochemical signals from the cells within microfluidic chips. The assay result is from our nanobio expert. So it's not from the industry. So it's a kind of ISO TC229 nanobio study group members. So we are going to do the survey again, as the Steve mentioned, and we focus on the professional industry representative to get a more narrowed and defined area for standardization. I think we have to do it first. And then, but I'd like to hear from you today. So each area should be standardized in terms of nanobio. And then also, if we have time, I'd like to discuss a little bit about our scope. And then, as I mentioned to you, we have 4 area working group 1, 2, 3, 4. And then we have left over 2 area, diagnosis, imaging, and therapeutics. Should be covered by existing working group, or some we need more different working group, or should we work together with the ISO TC 276 bio people, because they very interested in nanotechnology, because the biotechnology is very broad, so they want to have some expert from outside, such as the nanotechnology experts. So they try new thing. But in nanotechnology, it's kind of platform technology. Nanotechnology is the kind of essential part. So we have to talk with the ISO TC276 all together, and then try to find which one is the best way in the future. That's about it. So I'd like to hear from speakers and audience. Yeah, Simon.
Thank you, Tay. It's been an interesting afternoon. When we started this, I thought, what areas will we actually want to look at? You're right that NBS is just a working title. We chose it not to offend anybody else in the ISO world. That's why we spent a long time talking about it. But to me, I thought, oh, we may move into nanomedicine. But the overwhelming feeling from these presentations I've heard this afternoon is diagnostics. Diagnostics, 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 with the amazing advances made in this country alone, and also we heard from Singapore. So to me, one of the big gaps is already, you have it on the slide there, diagnostics and, well, therapeutics as well. I think this will be an area, and I've just been looking at what I said earlier today, and thankfully, one of the things chimes. So I said, a specific challenge to nano and biotechnology standard development would be developing reliable characterization methodologies. And that, to me, might be one of the key areas for diagnostics, seriously, because we heard so many examples this afternoon about that. And that could be a key area in itself, particularly given the expertise that's around this part of Asia. So what struck me is the amazing advances, but also the acknowledgement in the presentations that we'd like to get help from standardization. I'm sure we'd have a lot of very interesting discussions, but to me personally, it's a personal view, not a TC229 view. I think diagnostics could be a key area for us to look at.
Yeah, I agree.
Got my retaliation in first.
Any objection? Agreed? Disagreed? Yeah. I am perfectly totally agree with you. Another thing is the in vitro diagnostic is quite free from the regulatory of agents, because you're not going to use your human body. So we are using just serum, urine, and tissues. So at least we don't intervene our body. So it's quite okay. So
shall I just
Ali.
make one comment? Actually, I imagine that the market relevance here should not be that highly emphasized, because this technology is emerging and is in much earlier stage than the other areas of nanotechnology, as you all know. And when it comes to the issue of interaction of nanomaterial with the body, I mean the safety issue, having a standard is much more important, because our scientists, our researchers are using this. Our students in the labs, you know, the companies. So I think we should not put much emphasis currently at this stage on the market relevance, but if there is a market, okay. So we should not, because some people may consider that as a, you know, limiting barrier to come up with some of the new work item proposal. See what is the market relevance of that. This is a completely new area, and we should give the chance for further development, and due to the safety issues, I think it's very important.
Yeah, I agree. Yeah. So yeah, in my perspective, usually if after the industry grows up and the standards follow the industry, but in nanotechnology or nanobiotechnology, it's kind of we have to go in parallel way, and then also everybody shouldn't concern about the safety also. Yeah, thank you. So, other suggestions? I'd like to hear from the industry, and then you promised you wanna, because the company should ask us, the expert, to make a good standard. But we want to know which area we should focus on, because everybody kind of busy guys here. Everybody's very good, the distinguished scientist in his area, her area. So we want to focus on which area we should standardize.
Okay, first I'd like to thank all speakers, inspiring greatly this afternoon. And from the point of view of IVD manufacturers, I'd like to suggest, give my opinions and simultaneously ask some questions. So the first one is, it is obvious. As you just mentioned, it is obvious and clear that many companies have been combining nanotechnologies and biotechnologies to advance. But it looks like standardization, it goes a little bit faster before the market, nanobio market is fully grown or matured. So currently the business and competition is still beneath the surface. So if the standardization goes first and reflect industry's opinions or directions, then I think that standardization is inclined to the directions of global leaders. So that's the worries from growing or startup companies. So I think, as Dr. Lee just mentioned, the standardization and market growth should go parallelly. And second one is, I think the standardization structure consists of government and also designers as ISO and TC committees, and the last one should be industries. So, considering just the national policies instead of standardization, I think companies should help or assist this kind of government activities. However, standardization also is a burden to companies. We have to prepare many numbers of documents, and they could bias when we export our products to other countries. So I'd like to ask you committee members, ISO policies accelerating or encouraging companies to participate and advance this kind of international standardization activities. Thank you.
Yeah.
Sorry, I didn't get your name, but I want to sign you up straight away. Yeah?
Yeah, from the body tech. Yeah.
Honestly, I'm not being trite here. It's people like yourself that we need to be involved in the actual working committees. Now, the issue there is people from small to medium enterprises take up a large proportion of the company workforce. So to let someone like yourself go for one day is an enormous commitment. So I don't know if you can get together with your colleagues from similar companies, but you're exactly the sort of people that we need participating. And if you can't participate, you could at least write down your needs. So it's very timely that Tay is going to ask industry what we need. I was brought in when I worked for GlaxoSmithKline because I work for industry, but I work for big industry. You obviously work at the sharp end, and you're exactly the sort of person we need the views on. Well, that's my opinion. Go on, Steve. You must have a view.
Thank you, Steve. Simon. Yeah.
No comment.
Come on.
Yeah,
I mean, anything. Yeah, so praise us, like.
Right. Well, I do think even if we focus on diagnostics or analytical techniques, once again, it's like if industry can let us know, and it does, and it can be not even if you don't have the time to commit as a small, particularly as a startup, you don't have time. But if you come back and saying, hey, our variability in analyzing this particular molecule or this particular cell interface, just letting us know it's like this is where we're having variability and we need to have standardization. Or when we publish papers, we see variation on how people interpret it and how they're doing the experiments. So, and I heard you actually loud and clear, as even for Intel, some of the standardization, and it's more government regulatory, you're right, it seems to be very burdensome paperwork. But we're trying to really work more at a level of what is the procedure that you need help in standardizing? And that has nothing to do with government regulations for intercontinental shipment of goods. It could be when I'm doing my experiment and I'm taking this test, how do I standardize that I know somebody else that's trying to replicate my work is going to get the same results? So we're very much at a very, I think it's more just one level above R&D, but it's like if you have a standard practice and you want to make sure everybody's doing it the same when you publish your paper, your works, that's really what we're interested in.
Yeah.
Stephen.
Yeah, thank you, Steve.
Hi, I'm Stephen Thomas from Australia.
Yeah.
It's one thing to do the kind of research that a lot of people in this room do, and it's all very useful and very good to do, obviously, but it's a quantum leap to translate research into product innovation. So I think there is a lot of room here to be asking industry, to be questioning industry about what kind of products they might be thinking about based on, community market research. I think we're all in agreement that we're all in this game together for the benefit of the community. So I think it is really important that industry do work very closely with us, because they're the ones that are kind of out there doing market research and looking to innovate, looking to design products for benefit in the community. So I absolutely agree with Simon and others that, we ought not to be producing standards in a vacuum, in isolation. There absolutely has to be that interface with industry for if our standards are going to be of any use whatsoever.
Yeah, thank. So actually, the Korean experts, we are trying to do the two items right now. One is in the working group 3, and one is the working group 4. One is related to the Professor Lee, the high throughput screening, and then the second one is related to the MECASYS, the DNA hybridization system. So in working group 4, so we'll see how it goes in the working group 3. And actually, we received the feedback from the two companies. One is from the the Mecasys, and the other from the Samsung Electronics. So we try our best in terms of nanobio, and then we'll see how it goes, and then try to find a way to make a good standardization related to the company, industry company. So far. Yeah.
Okay. My name is K.S. Lu from Malaysia. Actually, I got two humble suggestions. The MBS 2015 or the MBS New Study Group shall conduct another round of survey, comprehensive survey, capturing the latest, the current status of the activities carried out on different countries. For example, I like the one, even it's a symbol, but it's comprehensive, from Iran. So they focus on the activities carried out in Iran.
Yeah.
So our group need to combine. For example, in Malaysia.
Yeah.
Okay, what are the, we got a national mirror committee on nanotechnology as well as biotechnology. They got a national policy on nanotechnology as well as a national policy on biotechnology. So they got balanced representations. The number one, the government must be involved. This was the initiative. From the policy, they come up with the initiative. Then they invite the people, representative from the university, institution of higher learning. Number two will be the research organization. The research organization cover both basic and applied, fundamental and what you call applied research. So then the other third group will be the professional body. For example, I'm from Malaysian Senior Scientists Association. So all these people are retired, but still they are very active at the age of 55 earlier. Now our retirement age in Malaysia is 60. So you got a group, they call professionals. So it can be involved with the, what you call the science, different what you call discipline of sciences, and as well as technology, or even what you call engineering. So then the other one is called from the manufacturer, you call industry. It covers both the small, medium industry as well as the multinationals. You got joint venture from other company in Malaysia. We just call them manufacturers. It can be multinational or small, medium. Okay, in Malaysia, they also set up the initiative by the government. They call it what you call biotech corporate. So as well as nanotech, they also set up. So then they got program. So the last but not the least, then you got another group they just call standard user. You come up with the standardization. They actually basically they are consumer. Anyone, you are the users of standard. So we also got a Malaysian Association of Standard Users. So it can be anyone. You use a standard. So as I say, we cannot just focus on, it must be what you call a demand from the what you call the customer or the client. Then the supplier, the manufacturer will do it. That is called reactive. You need the proactive also. The professional in the university and the research organization. Different countries, they were set up. For example, in the case of Malaysia, they got 5 apex university. 5, They got 5 out of 60. They got 5. So they got a center of excellence. They set up on nanotechnology. So they're focusing not only on the research. It covers both fundamental and applied research. They also do development. And they also, the center is called, adding one more word, they call innovation. Right? The meaning is, it must be, whatever come out, must be commercial. Right? They become a pilot, then you must be commercialized. Right? So they got a center. A center is not only on the research, also in innovations. Right? Innovation is any new ideas. So as I say, I just put forward my two humble, suggestion. Right?
Yeah, thank you.
Our study group should gather those countries that are involved with the NBS. They should come out with a country report on what they are doing. Right? Who are the people that are involved?
I'll do it.
Okay.
Yeah.
So then the second one, I mean, you combine it in the directory. So from the coming meeting, then at least other than Iran, also Korea, the other country, we know, right? But not in war. So for example, from here, I enjoy the benefit of IS. The paper was Malaysia is ranking 23. At least Malaysia is in. Number 1 is China. USA is number 2. South Korea is number 4. India is number 3. Right? 5 is Germany, and Japan is number 6. This is talking about the publication, which you qualify for the, what you call the ISI articles. There may be many other articles which are not qualified, not written, cannot be written as ISI articles. Right? So that's my two humble requests. Thank you.
Yeah, thank you. Yeah. So after this symposium, I'm gonna circulate the request related to the each country's the nanobio product and company. So and then we're gonna come up with some data and then request from the industry in the Edmonton. I hope to get this kind of data. So I'll ask the experts in each country. So I talked to the India and then Iran and also Mexico. They couldn't make it this symposium, but they're gonna give us some information about the nanobio product and then company information, also the requests for the standardization. I think that this kind of data can be good for our direction. Okay, I think it's the
time for dinner.
Time for dinner. So if you have a last burning question or comments, yeah, sure.
I have a doubt whether we can have nanobio, and the huge market is waiting about the nano agro.
Agriculture.
Yeah. Nano agro, because huge market, nano nutrient for the plant body.
Yeah.
That can be added into the same thing.
Yeah. Actually, there are some the opinion about we should put the nano agriculture in our table, but it's not too many. So from Mexico and India and a couple of countries mentioned about the nano agriculture. So we're gonna talk about that one too. So if you wish to bring up the nano related to the agriculture product, and then we can talk about it in Edmonton meeting, right? So thank you everybody. We're gonna finish up the panel discussion here, and then thank you. So the Professor Yoon is gonna finish up the symposium.
Yeah. Okay. Thank you for the panel discussions, and I think all of us are starving. So, and the dinner is ready just across the hall in the Merak room. And I'm very thankful for all of the audience and speakers and panelists for the excellent speeches and all the patients for it's kinds of heavy symposiums. And please enjoy the dinner and have more discussions on the nanobio and standardization and industrialization. Thank you very much, everyone. Thank you.